Hello guys, welcome back to this channel. This is the second part of the cyanobacteria video. In this, we will be talking about reproduction and importance of cyanobacteria in biology as well as ecology. So let's begin. Cyanobacteria do not show any sexual reproduction. No sexual reproduction. Instead, they reproduce by vegetative methods. Okay, they also show asexual reproduction. Asexual reproduction. What is the difference between vegetative and asexual reproduction? In case of vegetative reproduction, any vegetative cell, any part of the body gives rise to a new individual. While as in case of asexual reproduction, a specialized cell or specialized spores are formed which germinate into a new individual. Okay. In case of vegetative, we have methods like fragmentation. There is, for example, a trichome is like this. Suppose this is a trichome of any species like anabina. Okay, some of the cells break off due to some mechanical or some other factors, and this broken down, this separated part, this fragmented part gives rise to a new individual. Okay, this is fragmentation. The other vegetative method is binary fission. Okay, you have a cell, you have a cell, this splits and forms two new cells. Okay, this is binary fission. Okay, then we have an important one, harmogonia. Let me write it here. Harmogonia formation. Harmogonia formation. Harmogonia is a small mobile filament which breaks off from the trichome and then, okay, and then goes away from the main individual, from the main colony, and gives rise to new individual. Gives rise to new individual. Let me show you a picture of a simple trichome of, say, anabina. These are vegetative cells. And there is some specialized cell. Let me use it, which is comparatively bigger than other vegetative cells. And there is another specialized vegetative cell, okay, which is referred to as akinete. This is an akinete, okay, and this is a heterocyst. Okay, as I said, in case of fragmentation, the trichome breaks. It is usually at this position near the heterocyst where this trichome breaks off and results in the formation of a new individual, new colony, or new trichome. Okay, so these are the methods of vegetative propagation or vegetative reproduction. That is fragmentation, binary fission, and harmogonia formation. Then talking about asexual methods, asexual reproduction. Okay, asexual reproduction. As I said, that asexual is that type of reproduction where some specialized cells are formed, which are also called as spores. Here in this case, the akinetes that I showed, let me retry it here. Okay, they're usually elongated. They have the deposition of some carbohydrates, which kind of makes them distinctive. This is an akinete. Okay, these cells are like resting spores. These cells are like resting spores. That means they differentiate under unfavorable conditions, and when favorable conditions come, these cells germinate under favorable conditions and gives rise to new trichome, new individual. There is also formation of endospores. These are the spores which are formed inside the cell. Okay, if this is the vegetative cell, then these spores form inside the cell. These are endospores, and these spores are very tough, hard, and allows the cyanobacteria, and allows these individuals to overcome again these tough conditions, unfavorable conditions, and when favorable conditions come, these again germinate and form a new individual. So these are the spores, but you should remember that these are non-motile spores. That means the cyanobacteria have non-motile spores. That means there is no flagella on these spores, which is found in some green algae. Okay, so this is about reproduction of these, and as I said, there is no sexual reproduction, and they reproduce by vegetative and asexual methods. Now we'll be talking about importance, ecological and biological importance of the cyanobacteria. First, we will look at what is the ecological importance of these small prokaryotes. Okay, so ecological importance. Okay, so first is oxygen revolution. Okay, as we already discussed it in the beginning of this video, that over 2.4 billion years ago, okay, cyanobacteria changed everything by inventing oxygenic photosynthesis, which released vast amounts of oxygen and paving the way for all aerobic life, including we people as well. So this was called as great oxidation event. Okay, so second, foundation of food webs. Cyanobacteria are called primary producers of ecosystem because they are crucial base for food chains in many aquatic environments. Tiny marine species like Prochlorococcus perform more than half of the photosynthesis uh, in the open ocean, and thus producing a lot of oxygen and nourishing marine life. Soil health and fertility. On land, cyanobacteria play a crucial role in stabilizing soils in deserts and degraded lands, preventing erosion and helping other organisms establish themselves. Okay, and they are primary colonizers in succession. And then they pave the way for other higher forms to establish themselves. The fourth is nitrogen fixation. Many cyanobacteria can fix nitrogen. That means they change this nitrogen into NH3. For example, you have nostoc. Okay, you have anabina. Okay, as you may recall that these nostoc and anabina have specialized cells in their trichomes, which are called heterocystis. Suppose this is a trichome of a nostoc. Okay, these cells, they are comparatively larger than the surrounding vegetative cells. Okay, heterocystis. The heterocystis have thickened walls to prevent the entry of oxygen. Okay, thick walls. Why they prevent the entry of oxygen? Because these cells contain nitrogenase, which brings us about the fixation of N2 to ammonia, which is then used by the symbiotic partner of these cyanobacteria. So this nitrogenase, this is sensitive to oxygen. Okay, nitrogenase is sensitive to oxygen. As a result, to prevent the entry of oxygen into these heterocystis containing nitrogenase, they form thick walls. As well as there is one more adaptation, which is there is no PS second, photosystem second, in these cells. Why? Because this PS second has oxygen evolving complex. Oxygen evolving complex, which breaks the nitrogen into oxygen plus hydrogen ions plus electrons. Okay, two water molecules are broken down into a molecular oxygen. So this PS2 is here eliminated to prevent this inhibition of nitrogenase by the oxygen that PS2 releases. These are the two modifications, that is thick cell walls to prevent the entry of external oxygen into the heterocyst, and elimination of PS2 to prevent the inhibition of nitrogenase by the internal oxygen produced by PS2. So this nitrogen fixation by these cyanobacteria plays an important role in the nitrogen cycle. and being the primary producers also change the dynamics of carbon cycle. Okay, there is one more that is that they are also involved in pollutant degradation like heavy metals degradation and some other toxic materials. Now let us discuss briefly some biological importance. Biological importance of cyanobacteria. First importance is their food source. For example, spirulina. Spirulina is a cyanobacteria which is used as single cell protein and is cultivated as protein rich food for both humans as well as for animals. In regions around the world, cyanobacteria are an important sustainable food source. Second thing is bioactive compounds. They are also kind of chemists because these cyanobacteria produce antibacterial, antiviral, as well as antifungal compounds. Okay, making them suitable for use in biotechnology and medicine. Then the third importance is bioremediation. Cyanobacteria can degrade pollutants, absorb absorb heavy metals, and help purify wastewater, making them critical agents for restoring ecosystem and improving environmental health. And then biofertilizers. The association of anabina with azolla is used as a biofertilizer because anabina converts atmospheric nitrogen to NH3 with the help of nitrogenase, which is present in their heterocystis. Okay, so they are also used in formation of bioplastics, production of bioplastics. Compounds like PHAs. PHAs are produced by these. PHAs are polyhydroxy, polyhydroxy alkanoate, alkanoate. Okay, they have similar properties to that of plastic and are produced by these cyanobacteria. Okay, so these are kind of beneficial aspects of these cyanobacteria for we people, for the environment. But there are some challenges, especially the challenge is of algal bloom. Okay, algal blooms or cyanobacteria blooms. Okay, what are algal blooms? Algal blooms means rapid growth, rapid growth or exponential growth of cyanobacteria under favorable conditions. Under favorable conditions. What are the favorable conditions? Warm weather, warm temperature, high nutrient, high nutrient, especially nitrogen and phosphorus. Okay, and good amount of light. So that's why these algal blooms happen in the summers and early autumn. And most of these algae are scum-like. Okay, these algal blooms are scum-like and happen in stagnant water, or slowly flowing waters. Common species in algal blooms are Microcystis, Anabina, and Planctothix. Okay, these species, these blooms produce cyanotoxins, which are neurotoxins, means they interfere with our nervous system. They are hepatotoxins, that means they damage our liver, and they are dermotoxins. These toxins are very harmful for we people, for the cattle, okay, and for the aquatic life as well. These algal blooms are intensified by current anthropogenic eutrophication of aquatic habitats. And there is one more thing that cyanobacteria also cause black band disease of corals. Okay, what is this black band disease? It's a consortium disease which is caused by a consortium of bacteria, and this happens when some cyanobacteria turn into heterotrophs or rather parasites as well. So that's it from today's video. I hope to see you in the next video. Till then, take care. Bye bye.